My name is Barrett, and this is my presentation, Sticky Situations, How Bacteria Pick Their Partners. I performed this research in Guillemin lab this summer. All right, in a healthy gut environment, bacteria exist in multi-species microbial communities. Pictured here is a section of a mouse colon. In blue are human cells, in green is mucus, and in the confetti color combo are all of these bacteria that are associated with this section of the gut. So the bacteria live apart from human cells and they're separated by that layer of mucus that's sort of protecting the human cells. So in the gut, bacteria sort of exist in one of two modes, as part of an aggregate or as planktonic bacteria. So in previous research, we have observed the association of both Aeromonas and Enterobacter in the gut of the zebrafish. So when Aeromonas is by itself, it tends to colonize a different portion of the gut than when Enterobacter colonizes the gut by itself in a monoassociation. Um, However, when the two bacteria are together, Enterobacter will localize to the same region as Aeromonas is in. And so this overlap is where Aeromonas and Enterobacter are both at the same time. And I believe the green is just Enterobacter. So we have also performed this same assay in solution in wells. So here Aeromonas aggregates by itself, and then Enterobacter is planktonic. However, when they are added to a well together, Aeromonas and Enterobacter actually participate in co-aggregation. So here's what that looks like. As you can see, there's good amounts of both red and green bacteria, and there's not many green bacteria sort of floating around in the black regions. So I thought this was super interesting, and I wanted to explore it more. Specifically, I wanted to explore Enterobacter and why it's participating in this interaction. So I wanted to reveal important components in Enterobacter that allow Enterobacter and Aeromonas to co-aggregate. And to do this, I needed to select for individuals in the population that could not co-aggregate any longer. And then I also needed to identify what the mutation actually was doing and if it was relevant to co-aggregation. So to do the first half of this, I performed an experimental evolution. And so this was paired with co-aggregation assay. So Aeromonas and Enterobacter are added to wells and given the opportunity to aggregate. After they've formed those clumps, planktonic bacteria are pipetted off of the top of the well and cryopreserved, and then those bacteria are used for the next generation. And so, of course, when I was doing this and pipetting the planktonic Enterobacter off, there's both Aeromonas and Enterobacter in the solution. So our Enterobacter was GFP labeled and also had gentamicin resistance. So to separate Aeromonas and Enterobacter from one another, I was able to treat with gentamicin and then grow those overnight again, and then boom, more Enterobacter, good to go. And so after we started to see sort of a marked decrease, that's kind of where we go off this continuous passages path and actually plate the bacteria and get a little bit more information. And so that's exactly what we did. It took about 7 passages to reach a state where we were comfortable and really saw that there was a loss of co-aggregation. And we plated those individuals. And so we got a couple different mutants, but the most notable were this EV1, which we're calling the mucoid phenotype, and this is EV7 here, but it's been since changed to EV3. So EV3, the volcano phenotype. And so what we really focused on for the summer and for this presentation was EV1. And so here's a picture of EV1 on the plate. So you can see the wild type colonies are the smaller colonies, and then the mucoid are these very large, very shiny colonies. So they very clearly are different. Um, and here's some more data sort of showing how there is a marked decrease in co-aggregation with these phenotypes. So this is what it looks like under fluorescent imaging. I think this is really awesome because it really just drives home like the Enterobacter are really not a part of this co-aggregate any longer. This is such a huge difference. And so this was with EV1, imaging of EV1. And so um, this was so exciting, and we needed to send EV1 off for genomic analysis. So that's what I did, and we got our results back, and we found a mutation in the RCSD, or the RCS regulatory system, and particularly for EV1 in this RCSD gene. We also actually, since presenting this and putting all of this together, we also sent in EV3, and it is also damaged or mutated in a part of the RCS system, however, not the same gene. So not RCSD. It's I believe in RCSC. So that's really cool, really interesting. So RCSD is a part of the RCS system. Right here, it's sort of this McDonald's looking protein, but it's in gray. And so it interacts with IgAA, and it is partially responsible for cap synthesis. So cap is a sugar, an extracellular sugar. And this was the specific region of the RCSD gene that was mutated. So we believe the interaction with IgA and RCSD was somehow messed with. IgAA is a repressor of this whole system. And because we saw a lot of mucoid, or that would mean that the bacteria is producing a lot of sugars, this really all aligns with what we're seeing. And so this was the first model that I had come up with this summer. And so I had kind of believed that potentially the extra cap, the extra sugar, was kind of making the shield around Enterobacter that was making it so that Aeromonas couldn't get close enough to whatever it was actually binding to participate in co-aggregation. Since then, and since doing more reading and just getting more familiar with what CAP is and the RCS system in general, I kind of have a different view of what might be going on. In this model, CAP could be what Aeromonas binds to, but because Enterobacter is producing so much of it and in this different manner, it's actually shedding the cap as it creates it as an exopolysaccharide. And this is something that has been viewed in literature with E. coli. So rather than keeping the cap associated at the cell surface, where Aeromonas would actually bind to it and then stick to them, they're just shedding it off frequently. So the Aeromonas might be binding the cap, but then it isn't actually keeping the Enterobacter with it. So really excited to work more with this. I think I'm going to continue with this project. I just want to say thank you so much. My lab instructor, PI, and my mentor were all really central to this, and also Caitlin Kowalski. She did some awesome imaging for me, all of those really cool fluorescent images. And then, of course, thank you, Surf. It was so fun to get the chance to do this summer. So thank you.